we are going to learn introduction to plant breeding. What is plant breeding? What are the fields of plant breeding? What are the activities of the plant breeding and important achievements of the plant breeding? Friends, as you know that plant breeding is an applied branch of botany which deals with the genetic improvement of the agricultural crops. As you know that plant breeding can be defined as art, science and technology of changing and improving the heredity of the crop plants as per the needs of the consumer. There are another definitions of the plant breeding is a genetic adjustment of the crop plant in relation to the service of the mankind. And as you know that plant breeding is considered as the current phase of crop evaluations that was reported by Simmons in 1979. Here one example is quoted that Brassica oleracea is a one crop from which several other crop has been developed depending upon the selections of particular parts of the plants. As you know that why plant breeding is considered as art, science and technology that we are going to learn today first. Plant breeding is considered an art. In earlier days, when scientific knowledge was not available, the birth of genetics was not started, the plant breeding depends upon the skills and judgment in selecting the plants from the different variety. That means that human imaginations, human creativity and skills are the important parts of the plant breeding, and that's why the plant breeding is considered as an art. At that time, we do not know the inheritance of the characters, what is the role of the environment in plant breeding, what are the basis of genetic variations in the plants, and that's why that phase, earlier phase was considered as an art. But now plant breeding is an science. As the knowledge of genetics and other related sciences progress, the plant breeding becomes less of an art and more of the science. The modern techniques of crop improvement develops on sound scientific basis only after the rediscovery of Mendelian laws of inheritance that was discovered by Cornish, Testermarks and Hugo de Vries. So modern plant breeding mainly based on Mendelian principles of genetics. As you know that plant breeding is also techniques of development of hybridization and crossing is considered as art, but without knowledge, scientific knowledge of genetics, these techniques of hybridization is not going to play any role in the plant breeding. So now plant breeding is more a science and less of the art. There are 4 basic principles involved in the science of plant breeding: formations of the hypothesis, experimentations, observations, and finally we can give the conclusions based on our observations. Here one slide is shown that disease resistance and better flavor from the 2 different variety can be combined in one variety by the techniques of hybridizations, and that is the role of the science. Nowadays, plant breeding is also considered as a technology. Technology refers to the development of useful commercial product or a service involving the scientific principles and human skills. In plant breeding, the ultimate aim is to develop superior variety or a hybrid for commercial cultivations in relation to the existing variety, and nowadays we can use the techniques of genetically modified crops, marker assisted breeding as well as the speed breeding as a technology also. Now this slide shows you how the plant breeding has been progressed from art, science and technology of development of genetically superior variety in terms of economic utility for the benefits of the mankind, that start from the domestications, then selections and rediscoveries of the Mendelian principle leads a science, then it will be covered the hybridizations and mutations breeding and genetic modifications of the crop plants. But latest technology lead to the development of plant breeding as a technology using the GMO and marker assisted selections. Friends, as you know that there are 3 important fields of the plant breeding. One is a plant genetic resources, or it is popularly known as germplasm. Second, breeding techniques includes general breeding methods as well as the special breeding methods. So this is a very important part and core heart of the plant breeding. And third part of the plant breeding covers the seed production techniques. So ultimately we can give the variety or a farm hybrids at farmers' fields at reasonable cost, timely as well as the quality seed productions. Germplasm is a sum total of all the genes available either in cultivated form or in wild relatives for a particular species, and here one seed bank is shown to you that will reflect you about how the germplasm is important for the plant breeding. Without germplasm collections, breeder cannot do anything. So first and foremost important steps in plant breeding is the collections of the plant genetic resources at the research stations. Now breeding techniques. There are two breeding techniques. One is the general breeding methods that can be routinely utilized for the plant breeding experiments. And second, special breeding method that can be utilized on the special purposes. For example, selections, hybridizations, that are the routine techniques we are using as a general plant breeding method, but on exceptional cases, we are using marker assisted selections or a mutations breeding to rectify the defect of specific variety, and for that we are using the special breeding methods. And these breeding methods, friends, differ in self-pollinated crops, cross-pollinated crops and clonally propagated crops. And once you can achieve a variety, then you need to have a seed productions of these techniques by way of the seed productions. That is also a different classes that start from nucleus seeds, breeder seeds, certified seeds, and that will reach to the farmers to develop and cultivate that variety for the large scale. As you find that after the field of plant breeding, the third is the important aspect, and that is also considered as the industries also. So plant breeding is also an industries. Plant breeding can be viewed as an industries where improved variety or any hybrids can be produced on a large scale, and for that there are three important phases of plant breeding. One that is called as the research and development phase. It includes the collections of the germplasm, evolutions of the germplasm, and finally we are going to select some varieties. Then once the variety is selected or hybrid is selected, then we have to multiply that unit on a commercial scale, and once we achieve it, then we have to do the processing and packaging of that seed material so that this quality seed can be supplied to the farmers and a maximum benefit. As you friend know that before we are going to learn about the activities of the plant breeding, why plant breeding has to be studied. As you know that agriculture productions is increasing day by day. Simultaneously, the human population is also increasing. As you know that our agriculture productions in the year 1950 is 1582 million tons, which was 230 million tons in 2010. That means that 27.87% increase in the area and 255% increase in the yield become possible. And nowadays India becomes self-sufficient in food grain due to the harvesting the fruits of grain revolutions. But according to the well-known population geneticians, population is increased at a geometrical rate, while the food supply is increased at an arithmetic rate. And at present, the population of India is growing at a very alarming rate, 2.5% per year. And this make it necessary that food productions should also be increased either at least at the same rate or even at the faster rate in order to improve the nutritional status of the masses. And here you can see that India is the ranked first in the populations, while in food productions due to the harvest of fruits of the green revolutions, we are achieving self-sufficiency in case of the food grains. Now friends, as you know that what are the ways and means by which we can increase the food productions and increase quantum and better management of the inputs such as fertilizer, irrigations, plant protections measure and various cultural practices can be on one hand, but it can be increased up to some extent. While in case of the improved variety, we can increase per day productivity or per unit productivity, and that will be the aims of plant breeding. No doubt, net crop area can be also be increased to some extent, and that can be possible with help of the double and multiple cropping systems. But the high yielding, development of the high yielding variety or hybrid is the most important aspect of plant breeding by which we can increase the food production. So food productions can be increased by way of development of high yielding varieties as well as the hybrids. Now we are going to learn about the different aims and objectives of the plant breeding. Plant breeding objectives can be various and it can be modified from crop to crops also. So plant breeding aims to improve the characteristics of the crop plants that can become more desirable agronomically and economically. Thus the main objective of the plant breeding is to develop the improved varieties or hybrids of the crop plant that will be commercially successful at a farmer's level. So first and the foremost objective of the plant breeding is the higher yield. Most of the breeding program aim at higher crop yield, and yield product or a final product maybe depends upon the crops. This can be achieved by developing more efficient genotype. For example, Dwarf varieties of the wheat and rice we have developed, and that can be achieved a food sufficiency in our country like India. Now development of the hybrid varieties of the maize, sorghum, bajra, cotton and castor also lead to higher yields, and you can see that we have achieved this within a short period of the times also. Second objective is the quality improvement, and improved quality vary from one crop to another crop. For example, grain size, grain color, milling and baking qualities in wheat. Similarly, cooking quality in rice is very important criteria. Similarly, malting quality in barley. You can find different size, color and flavors of the fruits. Even a shelf life of the fruit is very important. Keeping qualities of the vegetables is also important. Protein content in cereals and legumes, lysine content in the cereals, and methionine and tryptophan content in pulses are the very important objective under the quality improvement program. Then the third important objective is the biotic resistance related to disease and insect pest resistance. Resistance variety offers a cheapest and most convenient method of disease and insect management. In some cases, they only offer a feasible means of the control. For example, various kind of the rust. Resistance variety has been developed in wheat. Resistance variety not only increase the productions but also stabilize the productions, and that is very important in our country like India. You can see that from the rust resist susceptible variety, with help of the breeding, you can develop a rust resistance variety. Similarly, insect resistance variety can also be developed with help of the various breeding techniques. Now the fourth important objective is the change in the maturity durations of the crop plant, and it has permitted the new crops and extended the new crop area due to the maturity durations. Development of the wheat variety for late planting has permitted the rice, wheat rotations in the country. Thus the breeding for early maturing variety or variety suitable for different date of the planting are an important objective in many cases. Desirable agronomic characters is also one of the important criteria, because we are thinking that modifications of the crop that will lead to some criteria. Modifications of agronomic characteristics such as plant height, tillering, branching or trailing habit is also desirable. For example, dwarfness, as you observed in the first figure, the dwarfness has been achieved associated with lodging resistance, fertilizer responsive, and we can harvest a good crop. Trailing habit in tomato lead to higher productions level, and that can be achieved from the second slides. While the tillering is an important objective in cereals, and you can observe that in rice that can be achieved in case of the rice also. Then many variety cannot be grown under the different photoperiods and different thermoperiods, and for that breeders play important role to develop the photoinsensitive and thermoinsensitive variety. Development of the photoinsensitive and thermoinsensitive varieties of wheat and photoinsensitive rice variety has permitted the cultivations in the newer area. Now I will give the examples of one where the rice is now cultivated in Punjab, while the wheat is a major rabi crop in the West Bengal due to photoinsensitivity and thermoinsensitivity of the breeding objectives. Then maturity, synchronized maturity is also a desirable objective in many crop plants where several picking can be converted into one picking, for example in green gram, pigeon pea, cotton, and that will lead to mechanized cultivations also. Another objective is the non-shuttering characteristics of the crop plants like in green gram, castor and wheat, where the shuttering is a major problem that can be achieved to convert the shuttering characteristics into non-shuttering habit, and that will be also possible to have a mechanized cultivations also. Determinate growth habit. Nowadays, all scientists are interested to develop high yielding variety with help of the determinate growth habit so that crop expansion, that means vegetative phase, can be limited and more durations is applied to the reproductive phase, and that can be achieved in case of the green gram, pigeon pea and cotton. Friends, dormancy is also one of the important plant breeding objective. Dormancy has two sides. In one side, dormancy is desirable, while on the other end, dormancy is not desirable in many cases. I will give the examples. Seed germinate before the harvesting if the rains comes at the time of maturity. For example, in green gram, barley, in spinach groundnut, it was observed, and you see that from the pod the plant is germinating. Under these circumstances, presence of dormancy for 15 days or 30 days is desirable to save the loss due to the dormancy periods. But in some cases, so dormancy periods may be break that will lead to achieve the crop. For example, when you harvest the groundnut in the summer season, that cannot be possible to grow in the next kharif season because of some dormancy. So in some cases dormancy is desirable, while in some cases dormancy is not desirable. Development of abiotic stress, especially moisture stress, salt tolerance and drought resistance variety is very important under the rain-fed area, and for saline soils also that can be possible to increase the crop productions. Nowadays you cannot increase the land, but whatever the land is available for cultivations, that can be used maximum so that that can be achieved with help of the development of the abiotic stress tolerant variety. The estimated salt affected soils in country may vary from 7 to 20 million hectares, out of which 2.8 million hectares are alkaline soils. So such soils can be utilized if you develop the abiotic stress resistance variety. Most of the areas are spread in the states of Uttar Pradesh, Haryana and Punjab. So that can be comes under the cultivations if you develop the abiotic stress resistance variety. Variety for the new seasons is a very important objective nowadays. Traditionally maize is a kharif crop, but scientists has now available it to grow throughout the year. Similarly, that can be happen in case of the permillet also. Mug is also grown as a summer crop in addition to the main kharif crops. The last but not least, the elimination of some toxic substance is very important to remove it for the safe consumptions for human being. For example, the popular examples, Kesari, Lathyrus sativus seeds have a neurotoxins BOOA that causes the paralysis. Similarly, brassica oil has a uric acid content that is harmful for human health. So removal of such kind of the substance lead to increase the nutritional status of the crops. All are at present talking about the nutraceuticals. So this aspect of the plant breeding is very important in case of the plant breeding also. Lastly but not least, the wider adaptability is an important objective of the plant breeding, because once the better genotype is available, that can be evaluated at large number of locations and number of years, and that will lead to stabilize the crop productions over the regions, over the seasons, and you can recommend that variety for the benefits of the farmers. So this is about the objectives of the plant breeding. Now different activities of the plant breeding we have to study in the details. So there are 5 important steps in the activities of the plant breeding. Creations of the variations is the most. Without variations, plant breeder cannot do anything. If variations is not available in a particular species, then that variations can be created by various means, then selections, then evolutions, multiplications and distribution to the farmers. Selections. What are the different ways to create the genetic variations? There are two important ways. One is the domestications and germplasm collections. These are the naturally available variations available for a plant breeders. Other means of the variations are available, but it has to be created by human beings. For example, mutations breeding, hybridizations, polyploid breeding, somaclone variations, and recently the genetic engineering are the ways and means to create the genetic variations in a particular crop species if these variations are not existing in available species. Then selection. There are two basic requirements for selections. One, variations must be present and variations must be transmitted from one generation to the next. Then and then effectiveness of the selections can be exercised. Here, out of these different crop plants, we are going to select two different plants that having a good yield potentials and that can be transmitted to the next generations will give you a result of selections. Once selected genotypes can be evaluated at different locations and years to have a wider adaptability, and that can be evaluated by various tests. There are two tests available at level. One is at All India Coordinated Improvement Program. It is tested for 3 years. Similarly, under the state trial, it is tested for 3 or 4 years, and finally the variety can be multiplied for distributions. So multiplications we can do at our wills, and that will be performed by seed certifications agency as well as the seed producing agencies and various other farmers, and once it is multiplied, then it should be distributed to the farmers and at a reasonable rate, timely and high quality seeds. So these are the 5 important, again I am going to repeat these steps: creations of variations, selections, evolutions, multiplications and distributions are the important activities of the plant breeding. Now, what are the important achievements of the plant breeding that we are going to learn today? First, semi-dwarf wheat variety. As you know that Green Revolution has led to the development of the semi-dwarf wheat and rice variety. The semi-dwarf wheat varieties were developed by Annie Barlow, and he is popularly known as father of Green Revolutions, and his associations at CMIT, International Center for Wheat and Maize Improvement Center at Maximo, he used a Japanese variety, Norin 10, as a source of dwarfing genes. And in 1936, ICR with help of the Dr. M.S. Swaminathan has introduced several dwarf selections from CMIT, Sonora 64, Sonora 63, Lermo-Rajo, and from this material, our scientist has developed the Kalyan Sona and Sonalika as a selections from this material, and that will lead to not sufficiency in food grains, but peace can be achieved in developing country, and as a result, Dr. M.S. Swaminathan is popularly known as father of Indian Green Revolutions. Semi-dwarf varieties of the rice were also developed from DGO gene, a dwarfing early maturing varieties of Japonica rice that is collected from the Taiwan. So Teaching native 1 developed in Taiwan and IR8 developed at Erie, Philippines was introduced in India in 1966 with the help of the Dr. M.S. Swaminathan. They will extensively grow in few years, but after this variety has been replaced by development of our own variety Jaya, Padma, Ratna and all these things, and that credit goes to the Indian scientists. So M.S. Swaminathan is popularly known as father of Green Revolutions in India. Third important achievements our Indian scientists has done, that is the nobilizations of Indian cane. Friends, you know that Indian cane, Saccharum barberi, was largely grown in the North India, but they were hardy but poor in yield and sugar content. On the other hand, tropical canes, Saccharum officinarum, has thicker stem, higher sugar content, but it cannot be performed badly in the North India due to the low winter climate. So C.A. Barber as well as T.S. Venkatraman and others at Sugarcane Breeding Institute, Coimbatore, transfer this thicker stem, higher sugar content and other desirable characteristics from noble cane to Indian canes, Saccharum barberi. And this is commonly referred to as nobilizations of cane, and that can be achieved. Nowadays, this nobilized cane can be grown all over the country as well as all over the world, and that credit goes to our Indian scientists. Development of the varieties of millet is also an important objective due to the availabilities of CMS line first in the maize followed by jowar and bajra will lead to development of various hybrid variety, and this hybrid variety have higher yield, early maturity as well as stabilize the yield, and various hybrid has been developed. For example, in maize, Ganga Safed and Deccans are the important initial variety, and jowar, CSH1, CSH2, 3, 4, 5 and many others has been developed, while in case of the bajra per millet, PHB10, PHB14 and JB 104, BK 156, these are the different hybrid developed by Punjab Agriculture University as well as the Gujarat Agriculture University in the past, and you can see that the photographs of these hybrids are very excellent, uniform and all these things. Then development of the hybrid cotton variety was done by our Gujarat scientist. The first hybrid variety of the cotton, H4, hybrid between Gossypium strains G67 and American Nectarines, was developed by Gujarat agriculture scientists at Surat research stations and released for commercial cultivation in 1970, and that credit goes by Dr. C.T. Patel. So, C.T. Patel is known as father of hybrid cotton. He used adioic methods of emasculations by practicing hand emasculations and pollinations. Similarly, our Dr. D.D. Gopani developed a Gujarat Castor Hybrid 3 as a first castor hybrid in the world, and that credit goes to our Gujarat scientist also. So, Gujarat is pioneer for the development of the hybrid variety in case of cotton, in case of castors also. Thank you very much for your patience.